1. Pinus seed cannot germinate and establish without fungal association. This is because it has a very hard seed coat. Explanation: Pinus seed cannot germinate and establish without fungal association. This is because it has very hard seed coat. Fungus associated with roots of pinus increase minerals and water absorption for the plant by increasing surface area, and in turn fungus gets food from plant. Therefore mycorrhizal association is obligatory for pinus seed germination. Question number 2. According to transpiration pull cohesion theory of ascent of scap, water is pulled upwards through the xylem. The cause of the pull is transpiration. According to transpiration pull cohesion theory of ascent of scap, water is pulled upwards through the xylem. The cause of pull is transpiration. Water is pulled upwards through the plants due to transpiration. Transpiration is the loss of water in the form of water vapor through stomata. The model that explains this translocation of water is called the cohesion tension transpiration pull model of water transport. Less than 1% of water reaching the plant is used in photosynthesis and plant growth. Question number 3. Which of the following characteristic feature is not applicable to parenchyma? Answer: cells are generally brick shaped, closely packed with intercellular spaces. Explanation: cells are generally brick shaped, closely packed with intercellular spaces. Is not applicable to the parenchyma. Parenchyma is a thin walled cell. These are packed compactly without the intercellular spaces. It is considered as a part of a simple per- permanent tissue. It forms the ground tissue and acts as a filler in various organs. It also forms the soft tissues of fruits. A generalized plant cell type, parenchyma cells are alive at maturity. They function in storage, photosynthesis, and as the bulk of ground and vascular tissues. Palisade parenchyma cells are elongated cells located in many levels just below the epidermal tissue. Gram-negative bacteria possesses peptidoglycan and an extra layer of answer lipopolysaccharide. Explanation: gram-negative bacteria possesses peptidoglycan and an extra layer of lipopolysaccharide. Both gram-negative and gram-positive bacteria have a peptidoglycan layer in the cell wall. Peptidoglycan is also called as mucopeptide. It is a polymer that is made up of sugars and amino acids. Gram-positive bacteria have thick layer of peptidoglycan. However, gram-negative bacteria have thin layer of peptidoglycan. Apart from this, gram-negative bacteria have an outer wall made up of lipopolysaccharide. Question 5. Which of the following is correct indication of the stage phase in the cell cycle? Synthetic phase. Explanation: synthetic phase is correct indication of the stage phase in the cell cycle. The cell cycle is the division of cycles into daughter cells through a series of events. In the cell cycle, there are two phases, M phase, and the interphase. Interphase is further composed of synthetic phase, gap phase 1 and gap phase 2. After the completion of the interphase, meiosis follows that is composed of series of events called the metaphase, anaphase, telophase, karyokinesis, and the cytokinesis that gives rise to two daughter cells. So the second event in the interphase is the synthetic phase, where the DNA gets doubled to prepare for the next mitotic division. Question number 6. Pollen grain can be stored for several years in liquid nitrogen having a temperature of answer minus 96 Celsius. Explanation. Pollen grains can be stored for several years in liquid nitrogen having a temperature of minus 96 Celsius. Flowers can be unisexual and bisexual depending on the presence of male and female gametophytes. Stamens mainly contains two parts, anther and filament. Anther is a bilobed structure that consists of microsporangia. Microsporangia develop further and become pollen sacs. Septum provides nourishment to the pollen grains. Microspore tetrads are formed and under meiotic division to form pollen grain. Pollen grains have two-layer exine outer layer and intine inner layer. Exine cannot be degraded even at high temperature and strong acids and alkalis due to the presence of sporopollenin. This layer provides protection to the pollen grains from adverse conditions. It helps to preserve pollen grains and prevent them from getting denatured for a long period of time. Various crop breeders store pollen grains and maintain their viability by providing specific environments. Pollen grains store at a low temperature. Preserving pollen grains at lower temperature, that is minus 96 Celsius in liquid nitrogen, is known as cryopreservation. Pollen grains are prevented Pollen grains are preserved at low temperature for a long time by reducing the growth rate of the cells. From the above information, we have reached the conclusion that pollen grains can be stored in a viable condition for many years in the liquid nitrogen at minus 96 Celsius. Question number 7. Which of the following options do not belong to acoelomites? Answer. Their mycelium is cyanocytic. Explanation. Their mycelium is cyanocytic does not belong to acoelomites. Acoelomites are fungi that are commonly known as sac fungi. They constitute the largest phylum of the kingdom fungi. Some of them are saprophytes and some are also parasitic. Various coprophilous species of acoelomites are also found all over the world. These group includes members like Penicillium, Aspergillus, clavis, clavis, and Neurospora. They undergo asexual reproduction via asexual spores, which are produced exogenously, such as conidia produced on conidophores. They include both unicellular and multicellular forms, and they possess separate hyphae.